Biased agonists act at G-protein coupled receptors. These receptors are important because they are targets of around a quarter of all drugs currently marketed to treat disease. Biased agonists can signal through one or more pathways, which they activate upon binding to a G-protein coupled receptor. In this way, they show functional selectivity for particular signaling pathways. This differs from the classical way of thinking about G-protein coupled receptors, where different agonists acting at the same receptor produce the same effects, but with differing potency and efficacy. Biased agonists reveal a more complex story, with agonists varying in their capacity to activate different pathways. To appreciate the concept of biased agonism, you need to first understand how G-protein coupled receptors were classically thought to work. G-protein coupled receptors are membrane-spanning proteins comprised of three different subunits. In other words, they are heterotrimers. The receptor binding site for agonists is in the main subunit projecting into the extracellular space. It is coupled to a G protein comprising an alpha subunit and a beta-gamma subunit on the cytoplasmic side of the membrane. When the receptor is inactive, the G-alpha subunit is bound to GDP. The binding of an agonist to the receptor initiates guanyl exchange, in which the GDP molecule on the alpha subunit is exchanged for GTP. This activates the G protein, allowing G-alpha and G-beta-gamma to dissociate from the receptor and go their separate ways so they can interact with their molecular targets in the cell. These targets regulate the activity of different signaling cascades. There are several different G-alpha subunits that affect different signaling cascades. Typical ones are G-alpha S, where the S stands for stimulation of adenylyl cyclase, and G-alpha I, where the I stands for inhibition of adenylyl cyclase. G-alpha Q11 stimulates phospholipase C activity, and G-alpha 1213 stimulates the small GTPase protein known as Rho-A. Modulating the activity of these enzymes will also affect the activity of other enzymes further downstream in the signaling cascade. Kinases that are stimulated downstream of G-alpha activation act back on the receptor protein in the cell membrane to add phosphate groups. There are also G-protein coupled receptor kinases in cells that can directly phosphorylate the receptor protein when it is in its active state. When phosphorylation is caused by kinases in the cascade activated by G-alpha, it is called signaling-mediated phosphorylation. When it is a direct effect of receptor kinases, it is called signaling-independent phosphorylation. Regardless of how phosphorylation is brought about, it initiates the desensitization of G-protein coupled receptors. What that means is that in addition to stimulating cell signaling, the binding of an agonist to a G-protein coupled receptor can also cause the receptor to desensitize so that it stops responding to the agonist. Once phosphorylated, the receptor becomes a target for beta-arrestin, which binds to the phosphorylated receptor and disrupts its interaction with the G protein by obstructing the G protein binding site. Beta-arrestin then primes the receptor for clathrin-mediated endocytosis, which removes the receptor from the membrane and internalizes it into the cell in an endosome. The receptors may then be dephosphorylated and sent back to the membrane, or targeted for degradation in lysosomes. Until just over 20 years ago, that was how G-protein coupled receptors were thought to work. Appreciation that their mechanisms are more complex began with the discovery that beta-arrestin interacts with additional signalling pathways that contribute to the effects of a G-protein coupled receptor agonist. But these effects are independent of the G-alpha or G-beta gamma subunits. While many agonists of G-protein coupled receptors behave as I have just described, some can activate the G-protein without stimulating phosphorylation of the receptor. So these agonists remain active for long periods without desensitizing the receptor. At the other end of the spectrum are agonists of G-protein coupled receptors that stimulate phosphorylation of the receptor by G-protein receptor kinases without activating the G-protein. That enables the recruitment of beta-arrestin, which desensitizes the receptor and stimulates its internalization, as well as signaling pathways downstream of the recruited beta-arrestin. So, biased agonists can stimulate either of two modes of signaling, preferring one over another. Agonists may be perfectly biased to activate the G-protein without stimulating the beta-arrestin pathways and receptor desensitization. Other agonists may be perfectly biased to activate beta-arrestin signaling and receptor desensitization without activating the G-protein. An example of a G-protein biased agonist is oliceridine, a mu-opioid agonist that is used to treat acute pain. The analgesic effect of opioid drugs is caused by stimulation of the G-protein, while stimulation of the beta-arrestin pathways is responsible for their side effects of respiratory depression, nausea, and constipation. Since oliceridine is biased towards the G-protein, it produces the good effects without the bad. An example of a drug biased towards the beta-arrestin pathway is TRV-027, which is under investigation for heart failure and COVID-19. It acts at the angiotensin type 1 receptor, which is the target for angiotensin receptor blockers used as first-line drugs to treat hypertension. Agonists at the AT1 receptor can increase contractility of the heart muscle and prevent apoptosis by stimulating the beta-arrestin pathway. These are desired effects. But stimulating the G-protein causes vasoconstriction and salt and water retention, which are unwanted effects. The bias of TRV-027 towards the beta-arrestin pathway means that it should produce the good effects without the bad. In fact, while TRV-027 is bound to the receptor, it prevents endogenous angiotensin from binding and stimulating the G-protein. So it provides additional benefit by acting as an antagonist of G-protein activation. While these drugs show strong bias to one or other pathway, most agonists lie somewhere in between. They show imperfect bias, activating both pathways to various degrees.